Okay, welcome back for the last review video for the AP Biotest. This is going to be covering Mendelian genetics and non-Mendelian genetics. Let's just say genetics. This is kind of the run sheet for the different stuff we have to talk about. So we'll go through some of the stuff that you should know, and then get into some of the stuff that they will probably see. And I'll just tell you from the beginning, you're probably going to have to know Mendelian genetics, but you're also probably going to have to know sex length. Those are probably, and blood. Those are going to be the three most important things for you to know going in. So, just as sort of a refresher, most people probably, most of you should know this. At least this beginning stuff. So you remember, Mendelian genetics has the concept of dominant and recessive. Okay, if something is dominant, it is the trait that shows, and if it is recessive, it is the trait that only shows if there's only recessive. And we denote those with a capital letter and a lowercase letter. Now, these are going to be your genotypes are going to be the letters, whether you're dominant or recessive, and the phenotype is going to be whatever that person looks like. So let's say we're talking about, I don't know, let's say we're talking about hair. So let's say this is brown hair and blue hair. Blue hair is not a thing. Let's say blonde hair. Okay, so this would be our genotype, and this would be our phenotype. Okay, the phenotype is what you see. The genotype are the letters, which you don't see. Okay, and from these we do crosses to sort of predict what different genes are going to be passed down and what the offspring are going to look like. And based on those predictions, we can determine which traits are dominant and which traits are recessive. So, we have the generic Punnett square where we can cross two parents. This is a heterozygous cross. We're crossing two heterozygotes. This is also what's called a test cross. Okay, a test cross you can do to determine what traits are dominant and recessive. So let me give you an example. Let's say, Mendel's actual example was done with pea plants and flowers. Okay, so he's got two plants. Okay, let's say, so I don't remember what the actual color was. Let's just say we've got two plants. These are plants. Look at that. Dang, I'm so good. Okay, so we've got two plants. We have a purple one. Look at this. I can't even draw inside the lines. So we have a purple one and we have a white one. Okay. Now, you as a scientist, you don't know which one of these is dominant or recessive. You don't know what's going to happen. All you know is that you got a purple one and you got a white one. Okay. So what you can do is you can cross them together. Okay. This is what's called your F1 generation. This is your first generation. Okay. Actually, that's not true. This is your parent generation. And so we don't know. All we know is we have a purple flower and a white flower. Okay. So what we can do is we can cross them. So let's say we cross them and we get the offspring from these flowers. And all of the flowers that we get are purple. Okay? 100% of the flowers that we get are purple. That's going to be our F1 generation. Okay? So this is the generation that we have right after our parents. So what that tells us is that purple, in this case, is the dominant trait, and white is the recessive trait. Okay? Now, that tells us which one is dominant and recessive, but if I wanted to know which ones, if I wanted to know what the genotypes of these plants were, then I would have to do a test cross. Okay? Now, we know the genotypes of our parent generation. We know that this one has to be homozygous dominant because all of our kids, all of our F1 generation, had the dominant trait. We know this one has to be homozygous recessive because all of our kids had the dominant trait. If this was heterozygous, if this was a lowercase letter, then we would have some purple and some white, but we didn't. We're saying we had 100% purple, which means that we had homozygous dominant and homozygous recessive. So what that means is that we know that these guys are heterozygous. Okay, and from there we can cross them to determine the actual ratio. Okay, and that is what is called a test cross. A test cross is when you cross two heterozygous individuals, and if the expected results match what you get. So from this, we would expect 75% of our offspring to have the dominant trait. 75% would be purple, and then 25% would be white. And so if we breed 1,000 flowers and we get 750 purple and 250 white or close to it, then we've done a successful test cross. And we know that this trait matches what Mendel predicted, that there's dominant traits and recessive traits, and that basically the gene that codes for these is just the one trait, okay? And that's really nice because it's really simple, okay? But what we found as more and more scientists have studied genetics is that a lot of genes are not that simple, okay? So this is sort of the basis for Mendelian genetics, okay? But we also have different kinds of genetics. The first one we'll talk about is incomplete dominance. Now, incomplete dominance is when you don't... Let me just pull up a picture. So incomplete dominance is when we don't have our two traits. Like Mendelian genetics is based on that this trait is completely dominant over this trait. So in these individuals that are heterozygous, the dominant trait is going to show over the recessive trait. So purple is going to 100% show over white. We're not going to have a light purple. We're not going to have purple and white spots or whatever. No, this whole thing is going to be purple because the purple trait is dominant over the recessive trait. But an incomplete dominant trait sort of says the opposite. An incomplete dominant trait says that those two traits are going to, in fact, mix together. So what we can see here is our red flower and our white flower are going to mix together to form a purple flower or a pink flower. So with incomplete dominance, we get a mix of two traits, okay? We also have a codominant, and codominant is we don't get a mixing, we get both traits equally, okay? So like here, we have a blue bird and a white bird. That gives us a blue and white bird, okay? The main difference with codominant is that both of our traits, we have two traits that are dominant, okay? So in this example, they're using C, so let's just use C, and let's change that to blue. So we have a capital C, but there's two, this is our dominant C, and we have our lowercase c, okay? But there's two kinds of capital C's. We have the dominant trait for blue, and we have the dominant trait for white, okay? And then we have the recessive trait. The recessive trait could be something else. Let's say, just for argument's sake, let's say the recessive trait is purple, okay? So what that means is that if we have a child, if we have a bird that has both of these traits, both blue traits, that bird is going to be blue. If we change that to, now we have a bird that still has two dominant traits, but now they have a blue trait and a white trait, that's going to give us a blue and white bird, okay? If we change this to they have a blue trait, and let's say we have that recessive purple trait, this bird is going to be blue because the dominant is going to win. It's going to show over the recessive trait. Okay, so when we say a gene is codominant, it's only going to be codominant with the other dominant trait. Okay, so here the blue and the white are going to be the codominant ones. So you could have a blue and white bird. There is no possibility of having a blue and purple bird or a white and purple bird because purple is recessive. The only way we could have a purple bird is if we had two recessive traits for purple. That would give us a purple bird. But if we had this recessive trait right here, if we had this recessive trait with either one of these dominant traits, the dominant one is going to show. Okay, so that's a little bit different of codominance between incomplete dominance. Okay, incomplete we get a mixing. Codominance you get both traits. Okay, but you have to understand that there is still the possibility of a recessive allele. Okay. And that kind of leads us to blood, because blood is an example of a codominant trait that we see in all people. Because last I checked, everybody has blood. So here's kind of how it works. So notice we have two dominant traits. Now blood we denote with an I. Okay. So we have two dominant traits. Okay. So with blood, we have the dominant trait is denoted with an I, and we have two dominant traits. You could have an A, or you could have a B. So both A and B are dominant. Okay. So it's possible that if you have A blood, you could have both dominant A traits, or you could have one dominant A trait with the recessive lowercase I. Now, it doesn't show it on this chart, but the recessive lowercase I is O blood. So you could be AO or AA, both of which would result in A blood type because the A is the dominant. The same thing with B. B is dominant over the recessive O, so you could have two Bs or one B, but either one of those is going to result in B blood. Now, if you have both dominant traits, if you have the capital A and the capital B blood type, then you are going to have AB blood. And if you have both recessive traits, that's going to give you O blood. Okay, so blood type is an example of a codominant trait where we have two dominant traits, A and B. We have a recessive trait, our O blood, and the only way that you can have O blood is if you have both recessive traits. That's what makes O blood so valuable and so rare, is because you have to have both recessives. Okay. Now, that kind of leads into a little more obscure, and I've kind of have them written here. So there's the idea of, let's talk about linked first. So linked traits are the idea that, because like all of this is dependent, right, all of the stuff that Mendel came up with, and even some of the non-Mendelian stuff is sort of dependent on that one gene relates to one trait, right? There's a segment of DNA that is responsible for, in my example, hair color, or in Mendel's example, flower petal color, or bird color, right? And that Mendel had what's called the law of, the longest thing that I have to write with a freaking mouse, the law of independent assortment, okay? And what the law of independent assortment states is that one trait will be passed on separate from every other trait. Okay, so it says that the trait for hair color is going to be a separate trait, is going to have no impact on skin color or eye color or height or intelligence or anything else, right? One gene is responsible for one trait, and it's basically you determine them totally at random. Okay, and to an extent, certain traits work that way. But what we found is that certain traits don't work that way. Okay. So for example, if we look here, what we know about DNA now is that DNA are combined into chromosomes. They're formed in chromosomes. And different genes are different segments of those DNA. And those genes relate to the genetics of what an individual looks like. So what we found is that certain genes, if they're located on the same chromosome, they're more likely to be passed on together if they're closer together, okay? Because remember, during meiosis, we get the crossing over event where these chromosomes mix up. So if two genes are really close together on a chromosome, they're more likely to show together than two genes that are really far apart on chromosomes. So for example, let's say that this gene right here is my hair color, and this gene right here is eye color, okay? Well, because they're so close together, we can say that those genes are going to be related most of the time. So, for example, generally speaking, let's say I know that a person has red hair, right? That red hair is their gene color. Well, I can pretty much say that their eye color is going to be blue eyes, because majority of the time, people with red hair have blue eyes. And the reason why that is because those genes are located fairly close together on the same chromosome. And that when the red hair gene is passed on, because the blue eye gene is so close together with it, they sort of go as a bundle, okay? So those are what we call linked genes. Those genes are linked together because of their location on the chromosomes. Whereas, let's say the gene for skin color could be on a totally other separate chromosome. So the skin color of a person and the hair color and eye color are, in this example, totally unrelated because they're on totally different chromosomes. But these two genes are linked together because of the location on those chromosomes. Okay. Now we see the law of independent assortment a lot with, so like we did a Punnett square here. You also need to know how to do dihybrid Punnett squares. Okay. And a dihybrid Punnett square also illustrates the concept of law of independent assortment. So like, no, that's not what I want. Yeah, that's what I want. Okay, so we can kind of see here, this is an example of a test cross in the dihybrid scenario. Now remember that a dihybrid scenario is where we're looking at two genes, okay? Di means two. So we could have an A trait and a B trait, okay? Now we're doing a test cross, which means, if you remember from the beginning, that's heterozygous. So we're crossing an individual that looks like this with another individual that also is heterozygous for two traits. Okay. Now this example the two traits that we're looking at are color and I guess shape. Where for the color we could be yellow or green, and for the shape we could be round or wrinkled. Okay. And those traits are denoted with A's. Let's say the A is for color, and let's say the B is for shape. Okay. Cool. So when we do that test cross, because we're looking at two shapes, we do the bigger Punnett square. This is the big 16-box Punnett square. Okay, now remember that when you're doing the 16-box Punnett square, you have to FOIL. Okay, so if for that test cross, what we're going to have to do is you're going to set up. Let's see if I can do this. I guess I don't need to do that, do I? Okay, that's really bad, but you get the idea. Okay, so this is our 16-box Punnett square, our very, very poorly drawn 16-box Punnett square. And remember that when you're doing two crosses, do not put one letter above each box. Do not do this. Do not go A, B, B. That's bad. That's wrong. I'm going to hate you for it. Don't do it. Okay. Remember, the first thing you have to do is you have to FOIL. Okay. You have to distribute. So you have to say, all right, we're going to do the first letter of this trait and the first letter of this trait. So we're going to go capital A, capital B. All right. And then we're going to do the first letter with the second letter. So we're going to go capital A, lowercase b. And then you do the second letter with the first letter, lowercase a, capital B, and then the second letter with the second letter, lowercase a, lowercase b. And then you do the same thing, right? It just so happens to be the exact same, so we do the same thing. And then from that point, you just do it like a normal Punnett square. You fill it in, right? You go across, you go down, and then you figure out, okay, how many of my individuals have the dominant A trait? How many are recessive A? How many are dominant B? How many are recessive B? And then you do it out of 16, Okay. Now, in the proper test cross, 9 out of 16 are going to be dominant A trait, and dominant B trait. 3 out of 16 are going to be dominant A recessive B. 3 out of 16 are going to be recessive A, dominant B. And then 1 out of 16 is going to be recessive for both. Okay. That's our test cross. Test cross for a dihybrid always comes out 9, 3, 3, 1 all the time, 100%. Okay. Now, remember that I also taught you how to do it a different way. And that is to do the individual Punnett squares, because most people are more comfortable with the individual Punnett squares than they are with this 16-box stuff. So if we have our two individuals and we say, okay, we can do this with any trait and any number of traits. So let's say this is one individual, and then our other individual is like this. Okay, so we're looking at 5 traits here. Now, if you want to draw out a giant ass Punnett square, you're more than welcome to. I don't want to do that. So what you can do is you can just do individual Punnett squares for these traits. So let's say these are our parents, and let's say we want to know what's the probability that we're going to have a kid that is simultaneously dominant for the B trait. Okay, so that is going to be dominant for the B trait and is going to be recessive for the E trait. Okay, well, we can just go through there and be like, all right, well, so the B trait is going to be this parent is heterozygous. This parent is homozygous dominant. Okay, so that's going to give us 100% that we're going to have a kid with the dominant B trait. And then we do another one, or let's do this. Let's do 4 out of 4, okay, because 4 out of our 4 kids are going to have the dominant B trait. And for the recessive E trait, this one's heterozygous. This one's homozygous recessive. So this one's going to give us 50% that have the recessive E trait, which is going to be these 2 squares, or 2 out of 4. Okay. So if we do some math and we multiply across, 4 times 2 is 8. 4 times 4 is 16. So that is 1/2. So we have a 50-50 shot of, with these 2 parents, having a kid that is dominant for the B trait and recessive for the E trait. Okay. So you have to do a little bit of math there. It involves fractions, but it's a little bit easier and way less time consuming than doing a giant 16-box Punnett square. Okay. Now, this is all examples of genes that are not linked, okay? Because what we found is that if genes are linked, when you do this math, we would expect 1/2 of our kids to have the dominant B and the recessive E. If we find it's more than 1/2, if we find it's, 80%, then those genes are probably linked, or they might be linked. Being linked could be a reason why we would get an answer different than what we would expect based on the Punnett square, okay? But that's how to do a dihybrid cross and how to do any cross that has more than 2 traits, because the AP Bio test will sometimes like to throw at you like 5 traits, 6 traits, 7 traits to see if you can do some quick math. Now, there's also the concept of pleiotropy, which we talked about in class. Now, pleiotropy is the idea that here's some other examples of like linked traits. Remember, we talked about this one, that the eye color is linked with the deafness trait. So this cat has a blue eye, and I know that because that cat has a blue eye, it's going to be deaf in this ear, because the eye color and deafness trait are linked together because they are close on the chromosome. Okay, this rat is fat because it is orange. The color of the fur and the metabolism traits are linked together. Now, pleiotropy is a little bit different. Pleiotropy states that not one gene is in control of one trait. Pleiotropy states that there are multiple genes in control of a trait. Okay, so and realistically, that if you think about it, that makes a whole lot of sense, because it's not just we don't have just like binary hair colors of you're either blonde or you're not blonde. There's varying degrees with which you can be blonde. There's varying degrees with which you can be brown hair. You could have really dark brown hair. You could have really light brown hair, or anything in between. And so pleiotropy states that we can have multiple genes that determine how dark to which degree that trait is. So, for example, hair color could be determined by 6 traits. The more of those traits you have, the darker the hair color you have. The less of those traits you have, the lighter your hair is. Okay, so it's not one trait that determines your hair. It's really 6 different genes that determine your hair. Okay, that's what pleiotropy is. And then sex linked. So sex linked is, in my opinion, the most likely thing you're going to see on the exam. And sex link works like this. So remember that you do a Punnett square, just like normal. Okay. But what you have to remember about sex linked is that, first of all, that males have an X chromosome and a Y chromosome. Females have 2 X chromosomes. Okay. And for the purposes of talking about sex linked, we're strictly only talking about the X chromosomes. Okay. And the reason why is because, get it? The reason why? I think I made that joke in class too. The reason why is because the Y chromosome is very, very small and doesn't contain a whole lot of genes. Okay. So most of the traits that are going to get passed on are going to be passed on along the X chromosome. The X chromosome is bigger. It has more genes. It has more DNA. So when you're doing a sex-linked Punnett square, the Y chromosome you don't do anything with. The X chromosome is sort of similar to how codominance works in that these X chromosomes can be dominant or they can be recessive. Okay, and you do it just like normal. You bring across your X's and your Y's first, because realistically, when 2 people have a kid, it's a 50-50 shot of having a boy or having a girl. Okay, now once we know it's a 50-50 shot of having a boy or girl, then we can determine what the chances are that those boys and girls have those traits. Right? We pass along our dominance. So this girl would be dominant for both traits. This boy would be dominant for the trait. This boy would be recessive for the trait. And this girl would be heterozygous for the trait. Okay. Now, what you need to remember about sex-linked is that because these genes are sex-linked, these diseases are much more likely to show up, or these disorders, whatever happens, is more likely to show up in boys, because the boy has to receive that trait only once. Okay. In order for the girl to get the trait, the girl will have to get the trait twice. So she has to be unlucky 2 times as opposed to the man having to be unlucky once. So if our disorder here is recessive, this guy, you have a 50% chance of having a boy, and then once you have a boy, you have a 50% chance of that boy having the disorder, assuming the disorder is recessive. But if you have a girl, there's no chance that the girl has the disorder, because the girl has to be unlucky twice, and that's not possible with this Punnett square. Okay, but what we can have happen is this girl right here can be what's called a carrier, where she doesn't have the disorder because she has the dominant trait, but because she has the recessive trait, it's possible to pass that on to her kid. Okay, so if this woman marries a guy that has the recessive trait, well, now that changes her possibility of having a kid. It increases the possibility that they have a kid with that disorder. Okay. So what you have to remember about sex-linked is that you can't just do it like a normal Punnett square. You have to only do it on the X chromosomes, and you have to set up your X's and Y's accordingly. Okay. And remember, you can always determine if a trait is sex-linked is if the mother has the trait. If the mother has the trait and a son has the trait, then it is a sex-linked disorder. Okay. Because in order for the mom to have it, the mom would have to be, like in our example, the mom would have to be like this. Okay, which means that both sons would have to be recessive, right? So the mom has it, the dad doesn't have it. Okay. Now, our Punnett squares for the girls are the same, right? Our Punnett squares for the girls, both of our girls are going to be heterozygous. So both girls are not going to have this disorder, but both boys are going to have this disorder. Okay, that is how you can tell if a trait is sex-linked. Here, our mother has it, so our sons also have to have it. That is 100% a sex-linked trait. Okay. Okay, so that covers Mendelian genetics, incomplete dominance, codominance, blood types, pleiotropies, linked traits, sex-linked traits, and the law of independent assortment. Okay, that covers most of what you're going to see from blood, from the genetic standpoint. You're probably going to see this linked. This material linked with what you know about DNA, what you know about transcription translation, and what you know about genes and gene regulation. Okay, so this information is pretty simple, especially because you've seen it twice now in pre-AP and in 7th grade, but it's how it all fits together with what you know about genes and what you know about DNA and protein synthesis to make a complete story. So what I would bet that you see is something like, they might ask you a transcription translation question, and then they might ask you a gene regulation question, and then they might ask you a genetic question back to back to back. Okay, so that just kind of is a refresher over the basics of genetics and the stuff that you're probably going to see. And things to keep in mind. Okay.